Today I'm going to explain you on Bohr Shift. So this Bohr Shift basically it will explain how a hemoglobin molecule is going to release oxygen in peripheral tissue and how it's going to bind to oxygen in the lungs. And in doing so, the hemoglobin molecule is going to carry protons and some amount of carbon dioxide from peripheral tissues, and it's going to release protons and carbon dioxide basically in the lungs. Let's understand how this hemoglobin is going to do this job. So let's see the peripheral tissue first. So here is the let's write peripheral tissue here, peripheral tissue. So as you all know that partial pressure of oxygen in peripheral tissue, that is PO2 in peripheral tissue, it will be between 20 to 40 millimeter of Hg, millimeter of mercury. So here is your red blood cell carrying, say I'll write hemoglobin here. This is a hemoglobin molecule. So it carries four oxygen molecules. Hemoglobin carrying four oxygen molecules. So as it reaches the peripheral tissue, so in the peripheral tissue, it's going to release these four oxygen. Generally, it releases two oxygen molecules. Whenever the partial pressure of oxygen is 20 to 40 millimeter of Hg, it basically half desaturation occurs. But for our understanding, let's do the 100% desaturation, means four oxygen molecules are released into the peripheral tissue. I'll repeat that. Under normal conditions, between 20 to 40 millimeter of Hg, there will be around 2 molecules of oxygen will be released, not 4. Okay. Just for our easier understanding, we are releasing all the 4 oxygen molecules into the peripheral tissue. So, because of the low partial pressure of oxygen, like 20 to 40 millimeter of Hg, so the oxygen unloading is happening. But it is not just the partial 20 to 40 millimeter of Hg partial pressure, which is helping in the unloading of oxygen. There is something else which will also help in the unloading of oxygen. So what is that? So basically in the peripheral tissue, as you know, like peripheral tissues are active tissues, so they will conduct TCA cycle, that is Krebs cycle. So Krebs cycle. So one of the byproducts of Krebs cycle is carbon dioxide. So this carbon dioxide is going to diffuse into the red blood cell. So let me write red blood cell here. So a little bit bigger, and here is the red blood cell. Red blood cell, so your carbon dioxide is getting into the red blood cell. Carbon dioxide gets into the red blood cell, whereas it is RBC. Now within the cytoplasm of red blood cell, carbon dioxide is combining with water molecules to form H2CO3. H2CO3. Okay, that is carbonic acid. This carbonic acid is spontaneously dissociated into H plus and HCO3 minus. H plus and HCO3 minus means protons and the bicarbonate. So these protons, basically they will go and protonate histidine residue. That's what these protons will do. So the protons will protonate histidine residue. They will go and bind to hemoglobin molecule. Basically, 2 protons will go and bind to histidine molecules in a hemoglobin, and that will help to release 4 oxygen molecules. 4 oxygen molecules will be released. So to get these 2 protons, let's balance the equation. So 2 carbon dioxide enters into cytoplasm, combining with 2 water molecules, and 2 carbonic acid will form 2 H plus and 2 HCO3 minus. 2 bicarbonates are formed there. Now what will happen to this bicarbonate? So bicarbonate is basically the carbon dioxide. So bicarbonate will be moving out. So this bicarbonate is coming out of red blood cell into the bloodstream. HCO3 minus is now, it is present in the blood, enters into blood. Now this is a negative charge which is coming out of the red blood cell to maintain the charge equality. So chloride is getting in. Whenever bicarbonate is coming out, chloride is getting into the red blood cell. This is called as chloride shift. Now your hemoglobin molecule in the peripheral tissue, it has released 4 molecules of oxygen for every 2 protons binding, and 2 bicarbonates are coming out. So This is how 90% of your carbon dioxide, 90% of carbon dioxide is carried back to the lungs in the blood in the form of bicarbonate. So 10% of the carbon dioxide directly it will bind to hemoglobin molecule. Particularly the amino terminal side of the hemoglobin molecule will be binding with carbon dioxide, that is only 10%. So 10% of your peripheral tissue carbon dioxide is carried by directly binding to hemoglobin, that is called as carbaminohemoglobin. 90% of the carbon dioxide is carried in the form of bicarbonate. Now let's take this RBC back to the lungs, because venous return will occur, your peripheral tissue, venous return. So it is basically going back to the lungs. So in the lungs what will happen? So here I'll write lungs. As you all know that CO2 of lungs is 90 to 100 millimeter of Hg. Now what will happen to your RBC? Let's write the RBC here in the lungs, red blood cell. Okay, so the hemoglobin. Now your hemoglobin, it is coming from peripheral tissue to the lungs. So within the lungs, so basically your hemoglobin is having these 2 protons, 2H plus, and also it is having carbon dioxide, CO2, carbon dioxide. Okay, and that's 10% of carbon dioxide, which is brought from there. So now the partial pressure of oxygen is 90 to 100 millimeter of Hg. So at this high partial pressure of oxygen, so your oxygen is going to get into the red blood cell and binding to hemoglobin. So now oxygen, 4 molecules of oxygen will bind to hemoglobin. So when the 4 molecules of oxygen will bind to hemoglobin, basically hemoglobin is undergoing a conformational change, and that gets into R state of hemoglobin, that is relaxed state of hemoglobin, R state of hemoglobin. Whereas here in the peripheral tissue, when the oxygen molecule is released and the protons are binding, hemoglobin is getting into T state of hemoglobin. Okay, anyway, so now your hemoglobin is getting into R state, and because of this conformational change, so the protons that are bound to histidine molecules, so they will be released. These protons will be released into the cytoplasm. Okay, now your bicarbonate, which is carried in the blood, here is the blood bicarbonate. So this bicarbonate, now it enters into red blood cell in the peripheral circulation. So the bicarbonate that is coming in the blood, now it is entering in HCO3 minus, enters into the red blood cell. At the same time, chloride is coming out. So this now balances the negative charge. Previously in peripheral tissue, what we have seen, when the bicarbonate is coming out, chloride was moving in. Now in the lungs, bicarbonate is getting into the red blood cell, chloride is moving out of red blood cell. Okay, what will happen to this bicarbonate? Bicarbonate will combine with these 2 protons and will form H2CO3, that is carbonic acid. Now the H2CO3 will be, let me increase the size of red blood cell here, so that I'll write the complete reaction. There is a red blood cell. H2CO3 will be spontaneously broken down into CO2 and H2O, water. Okay, now the CO2 will be diffusing outside, it diffuses out and it will be exhaled. CO2 is exhaled. This is how, and whenever the oxygen is inhaled, this is the inhaled oxygen, inhaled. We inhale oxygen, and that is the oxygen which is binding to hemoglobin. Carbon dioxide which is released from the red blood cell, we are exhaling it out. Okay, this is how oxygen is brought from the lungs and released in the peripheral tissue, whereas the carbon dioxide, which is the byproduct of Krebs cycle, is taken from the peripheral tissue and it is released in the lungs. This entire effect we call it as Bohr effect, or it is also called as Bohr shift. I hope you understood the concept. Thank you for watching.